after 9/11. I've worked with virtually all of the major vaccine candidates for biodefense purposes. I was at the tip of the spear in bringing the Public Health Agency Canada vaccine forward that we now call the Merck Ebola vaccine. I got Merck involved, and I got the money from the Norwegian government that funded the ring vaccination trials. And then I have also been right at the forefront of drug repurposing first for Zika. There's a number of papers relating to Zika and Zika pathogenesis and drug repurposing. Started a company that went bankrupt. I've done, I think, 4 different startups in my life. And because there was no funding for drug repurposing. And then since the beginning of this outbreak, I got a call from a member of the intelligence community that was in Wuhan that I've published with in the past, was in Wuhan during the 4th quarter of 2019. And he called me on January 4th of 2020 and told me that I needed to get my team spun up because of this new pathogen. So we started working on computational screening and docking of the entire library of licensed drugs. And that eventually led to my self-administering some of those when I became infected with the Biogen outbreak at the end of February 2020. And that led to the discovery of famotidine or Pepsid. I wrote the Northwell contract for that together with Jim Talton. Northwell screwed up those trials, but we've carried on. I've got multiple papers published or in press involving famotidine and then the combination of famotidine and celecoxib. And it's taken, the first of those papers was uploaded in July of 2020. It's been through peer review 3 times and still not been published. It's been approved in peer review 3 times and then pulled by the editors of Frontiers at the last minute, just like Pierre Corey's paper was. Mm-hmm. But those are the trials that we finally have FDA clearance to proceed on. So I'm one of not very many people that understand this whole spectrum of vaccines, pandemics, drug repurposing, pharmacology, and molecular virology.
Mm-hmm. So there's a new drug that is in clinical trials 3, I believe. It's called, I may be pronouncing it wrong, but Malone Pyrovir.
You're talking about the Merck product?
Yeah.
It's originally called EIDD-2801. It was initially funded by the crew that I work with at Defense Threat Reduction Agency. We tried to get it in as part of the formulations. We're using a multi-drug approach, and we wanted to include it as the antiviral in our combination. The other agents are anti-inflammatories, but Merck acquired it from Ridgeback. The same character that I was just referring to that works for the 3-letter agencies used to report to the Assistant Secretary for Preparedness and Defense, Bob Kadlec. He helped broker the Merck deal. EIDD-2801, there are, so the name, it has to do with the Emory Drug Discovery Group. Ray Shinazi is a famous member of that. So that's the origin of the original acronym. And there are others at Emory that feel that this is a highly inappropriate compound to be advancing because of potential reproductive toxicology issues that they're concerned about. We'll see how that fares. There's also, so this is one of the new hopes for a direct-acting antiviral. The other one is the Pfizer 3CL protease inhibitor. So when Tony Fauci kind of did his great pivot 2 weeks ago and suddenly started talking about, well, we need drugs that we can administer early and turn COVID into something like the common cold, he was only really thinking about these 2 agents, and he has no enthusiasm for a repurposed drug. At the DoD, fortunately, we're entirely separate from Dr. Fauci's control and oversight, and so we're able to pursue the science that we see as most appropriate. And we've been focusing on drug repurposing, as I said, from the beginning of 2020. And we actually in the trials, we had designed a 3-arm outpatient trial that would be placebo versus celecoxib and famotidine versus celecoxib, famotidine, and ivermectin. But the FDA created such a storm with their objections to including ivermectin. They put clauses in there that were so untenable. It would have taken us 6 months to a year to satisfy them. So we just dropped the ivermectin arm. But the data suggests that the triple combination is really potent. And surprisingly, the addition of, so we've been very laboratory focused in terms of, so the famotidine and celecoxib combination, even in, certainly in outpatients, but in inpatients, we can see a point of inflection in a lot of the key labs that are predictive of outcomes of COVID when we start administering drug. It's quite striking. We now can manage COVID using classical laboratory assays and make decisions about clinical management. One of the things that was intriguing about the addition, those with ivermectin, is that we saw much more rapid improvement in the leukocyte fraction in the CBC counts. And it appears that there is some benefit in terms of lymphocyte recovery. And but then there's the recent data out of Israel that I had thought was not going to be forthcoming. There's long been speculation that ivermectin might have some direct acting antiviral activity, and the new Israeli double-blind randomized clinical trial with only 55, I think is right around 50 or 60 patients, hit statistical significance and demonstrated that there is an antiviral component to the ivermectin activity. So, you know, we'll be testing famotidine and celecoxib because that's what we could get through the FDA, and that took us 3 months of negotiation. But I suspect that in the end, people will find that the triple might be even more useful. Thereby, I address that spectrum of pharmaceuticals. And so you saw the fluvoxamine data came in positive today. That's more news. Something like 38%, 35% protection in RCTs. Ivermectin in that same trial did not show efficacy, but they administered the ivermectin late and at relatively low doses. So a case could be made that that one was kind of set up to fail for the ivermectin arm, but the fluvoxamine definitely hit statistical significance. So we now need to add fluvoxamine as another agent that we already knew that. You know, I don't know how attuned you've been to the fluvoxamine story, but I'm in touch with Steve Kirsch, you know, almost hourly. He's the one that funded the George Washington studies, GWU. This is a different, so this is now the 4th study about fluvoxamine that's out there that I'm aware of.
That's great. You know, see, the watchers need to realize that there's more tools in the toolbox as researchers like yourself are investigating compounds to try to fight the disease, COVID-19, or, you know, the virus, SARS-CoV-2. There isn't necessarily the vaccine. And the way Fauci has been spinning it, it was Moderna from the get-go.
Yeah, well, you know why? That's the vaccine, right? That came straight out of the VRC. And Moderna was created by DARPA. So there's all kinds of angles here that I prefer not to go into, having to do with Bill and Melinda Gates Foundation and the tight relationships there and their lobbying, and you know, Zuckerberg Chan and Robert Wood Johnson and WHO and Facebook, and there's just a huge array of folks that are with money and power that have gone all in on vaccines as the only solution. And I think I'm a vaccinologist, but my assessment from the outset was that the risk of antibody-dependent enhancement based on prior work with coronavirus vaccine development was so great that, and the timelines to demonstrate a safe and effective vaccine were so long, and the risk that this was going to hit the states. Remember, I started in the beginning of January. I was docking compounds on January 11th after the seafood market, Wuhan seafood market virus sequence was first uploaded. I didn't focus on 3CL-Pro, which is what the Pfizer drug is inhibiting. It's a protease inhibitor. Rather, I focused on the papain-like protease because I knew that there was already a lot of candidates out there and a lot of attention on 3CL-Pro. And the Pfizer is one of many that are candidates for inhibitors of 3CL-Pro. These are both serine proteases, and serine protease inhibitors are notoriously nonspecific in the pharmaceutical industry and prone to dose-limiting toxicity and complications. So time will tell how that plays out, and I wish them the best. But with my group of pharmaceutical experts, we've always been very wary of the potential risks associated with focusing on a serine protease.
So the cocktail that you've been investigating, is that with or without the corticosteroids? For an individual that are...
So, matter of fact, we've got strong data that if you add dexamethasone, you kill people. No, I'm not kidding you. You can laugh. The paper is out there. We just had it provisionally accepted. I just got to make a couple modifications about how many people in the study group were smokers, which affects risk, and not always in a negative way. So they were not active smokers. They were recovered smokers, and that actually, because of the groups they were in, it actually makes our conclusions even stronger. But if your readers, or if you want to post a link, I can send you we put everything up on preprint servers right away because of the nature of the emergency. And so the famotidine plus celecoxib with and without dexamethasone paper is readily available on a preprint server, and it clearly shows we jumped from 0% case fatality rate in hospitalized COVID, WHO, 4 to 6, to something like 23 or 25% mortality in the presence of dexamethasone. We then verified, that's not generally understood by most docs, but the data supporting Dex is really quite tenuous. It took a lot of statistical manipulation to show statistical significance. It's in a very small cohort that it does reach statistical significance. It's very limited. It should not be used as widely as it's being used, if you're going to go fully evidence-based. There's overlap in terms of the mechanism of action with celecoxib. When you add the two, they are definitely more toxic and lethal. And this was verified in a platform trial called iSpy. That hasn't been published yet, that we also funded through DoD. We had counseled that they should not proceed with the trial arm in the presence of Dex, but they insisted that they do so because they considered it standard of care in they're basically a WHO 5-6 cohort. So this is under high flow oxygen or intubation. And so that is where Dex is indicated. They insisted that we had to have Dex on board, and they wanted to go ahead with the trial. And in fact, they verified that the trends were such that we would be, I don't know how to say this delicately, more patients would be lost on the triple combination. Their study also has a standard of care remdesivir, and I suspect that your viewership may be aware that remdesivir's efficacy is not quite what we were led to believe. And most would say that it doesn't support the license that it has right now, the authorization. In any case, that's a tangent. That's going down a rabbit hole. But yeah, stay away from the steroids if you're going to use famotidine and celecoxib. And it's a good thing, and you'll see in their paper, we've got a lengthy discussion in that one, in which I extensively quote Tony Fauci for an interesting review paper he did on Dex a few years ago. Dex is like a great big hammer to the immune system, and absolutely not. A case can be made that Dex is a great drug if you've got hospitalized COVID and you want to pump your numbers up by getting them out the door and transferring them to a extended care facility instead of having them die in your hospital to be a little bit jaded. But in fact, that's what goes on. Is there's a lot of manipulation of hospital case fatality rates by offloading patients to various triage options. And DEX is a great way to stabilize them, get out the door, and that is one way that it's used in some hospital environments.
That's very interesting. So let's pivot to the antibody-dependent enhancement and vaccine-associated disease enhancement. Now, what's your take on what's going on with Delta? I mean, from your observation, where you're sitting, and all the research that you're doing.
I see two hypotheses. So there's I just mentioned the Lancet paper that's just out that you haven't had a chance to read yet, which takes patients that were basically 2 months out from vaccination that are breakthrough cases and examines their characteristics. So there's no real solid control in the sense that you don't have Delta in unvaccinated or Delta in, there's kind of like three cohorts, right? There's vaccinated, there's previously infected, unvaccinated, and then there is naive unvaccinated. Right. Those are three key cohorts. And this only examines the one which is vaccinated at a time point. It's a slice of patients that are breakthroughs at a time point of approximately 8 weeks after vaccination. So basically at completion of dose 2. So this is peak for immune response in theory. And what they found was that in the patients who had these breakthrough infections, I think the number, and I don't have the paper here in front of me, so fact checkers, please don't skewer me. Forgive me for my elderly mind that is not a steel trap as it might once have been, but I think it was 20 plus fold higher levels of viremia than observed with previously with the alpha variant. And this is in the vaccinated cohort infected with Delta. So comparing historic data involving alpha infections in the unvaccinated to Delta infections in the previously vaccinated times 2 months prior, in those breakthroughs, they saw a huge increase in the overall titer relative to the comparator. But then what they found was the characteristics of the breakthrough cases was that they were the subset that had relatively low titers. So basically, it's not that the case is being made that it's not, the distribution of breakthroughs is not uniform in this cohort that is 2 months out after vaccination, but rather is skewed into the cohort of vaccine recipients that were relatively low responders. Why does this matter? What we're seeing in the Israeli data, and now UK and some of the other countries that are coming online, is that particularly in the Israeli case, Delta hit at about 6 months, 4 to 6 months after they finished their massive vaccine campaign. And in Israel, we had really good vaccine uptake. That's part of why Pfizer selected and did this special deal with Israel. And that has some interesting contract terms, I'm told, having to do with prohibited disclosure of adverse events that are collected. But yes, I have this straight repeatedly from Israeli scientists. It's not publicly disclosed. So Pfizer went into Israel because it's a very compliant population. And in fact, they got very good vaccine uptake. And then you may recall a few weeks ago, Pfizer announced that the vaccine was going to need to be boosted at 6 months because the durability was poor. And you may recall Dr. Fauci reprimanding them for making that statement. And then 2 weeks later, this became U.S. government policy. And they're now about to roll out revaccination dose number 3 at 6 months in high-risk elderly and immunocompromised. So that's pretty well an admission of this. So the thing about the Israeli group getting hit by Delta at around 6 months is that is just the window when they're starting to move into the majority of their population being in the waning phase or no longer effective phase of the primary vaccine that they received. And as you know, and I'm understanding that your viewership is a more sophisticated audience, the highest risk for antibody-dependent enhancement is in the waning phase because the slope is long. So there's kind of like two windows of high risk. If you can think of my intersection of these two straight lines of my hands, if you imagine the peak here is shortly after completion of 2 doses, the ascending phase of the immune response is fairly steep. And so the delta T in which you cross between the threshold of immune response necessary for protection and as you're climbing towards peak response, it's fairly short. It's very brief. And then there's a long declining phase. And so when you cross that window where you still have antibodies, but insufficient titer of antibodies and presumably insufficient titer of the most potent antibodies because of the waning phase, you have a much longer window of susceptibility where you still have antibodies around that can bind the virus, but you don't have enough that will neutralize